Hello, class. So, welcome to our, I don't know how many lessons do you have in the laboratory already, pero welcome sa laboratory setup and equipment. I think this is the second, right? So, let's see. Let's review how your molecular lab is set up and what are the equipments inside of it. So, yan ang pag-aaralan natin today at ang workflow. Yan. So, na-discuss ko na to, di ba? So, okay. So guys, number one na enemy natin sa molecular biolab is contamination. And this contamination, guys, there's a lot because we're now in molecular level, guys, hindi ba? So, ang contaminants natin, kaliliit talaga, hindi natin makita with our naked eye. We don't like nucleic acids into our specimen. So, every cell na lumilipad dyan, yan, may nucleus yun. Tapos sa loob nun, may nucleic acid, may ribosome doon, may mga RNA doon, guys, may mga enzymes doon. So, talagang ang raming contamination in a molecular lab. And as much as possible, we want to minimize it, right? So example pa lang sa PCR machine, isn't it that our PCR machine is like a photocopy of DNA? So the products, the amplification products is as much as 10 raised to 9. So siyam na 0. Yan. So kung ang 6 na 0, tama ba? And million, tama ba? Yan. So pag 9, trillion. Oh my goodness. So, ang daming guys na nucleic acid sa molecular lab. I think you get the point. And the sources are cross-contamination, the products, laboratory surfaces, ventilation, hair, skin, saliva, and the clothes of laboratory personnel. And we want to minimize these things. So that's why your your laboratory is organized in such a way to minimize it. Hindi ba we work from clean to dirty. Yan. So the clean areas, we maintain its cleanliness as much as possible, no contamination. And in the dirty area, yan. So dirty na talaga doon. Okay? So wag na natin ibalik yung, so sa mga dirty areas, wag natin ibalik sa mga clean areas. So correct handling of reagents, di ba? If you notice, our micropipette tips have filters. Yeah. So because aerosols are created upon our aspiration of reagents, so there's a correct way of handling them. And reagents are handled in a specific area only. Each room or area needs to be talagang separated, okay, with walls. And 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 each room, guys, has dedicated equipment. So if the pipettes are only in the pre-PCR room, it only stays there. Okay, so dapat may dedicated, may separate room, may separate equipment. So pipettes, kailangan ng pipettes sa lahat ng room. Dapat hindi siya nalilipat-lipat sa ibang area. Okay, meron tayong mga mini-fuge. Yan. So the mini-fuge in the specimen prep should stay there. It should not move to any other room in the facility. And hands must be washed and gloves and lab coats changed when moving between the designated areas. So we are vectors, or tama ba? Yun, we are vectors of contamination. That's why there are changing rooms. Yan, so as to limit the contamination. Grabe, no? Kailangan may changing areas. So let's learn more about the setup in the laboratory. So, my basis for this discussion is talagang AO, Administrative Order 14 Series of 2020. So this batas to, guys. So hindi ko inimbento to, okay? So we have a Filipino guideline. So based from WHO recommendations on how what are the requirements to be able to operate in a COVID-19 lab. So, namili lang ako ng ilan, okay? So, COVID-19 testing shall only be done in a DOH licensed COVID-19 testing laboratory. Wala lang iba. So, dapat guys, license ang laboratory by only, one and only, DOH. Okay? So, no underground laboratory. So, iba ang lisensya ng isang lab that is able to perform a COVID-19 testing. COVID-19 testing laboratory shall be a separate unit, okay? Because it's very prone to contamination. Strict adherence talaga to biosafety, biosecurity, infection prevention and control. And guys, COVID-19 testing laboratory shall be strictly prohibited from outsourcing examination. So, kanya nagbag yung RT-PCR mo, you cannot outsource it. You cannot let other laboratory test it for you. Yan. So, strict siya. Okay. Guys, we only use FDA-approved kits, yan, agents and devices. So, hindi tayo basta-basta bumibili lang sa mga online shops, kahit anong ahente, make sure that they are FDA-approved. COVID testing shall be in a separate, okay. Ito. Strict adherence, ano ba yan? Ito na. Supervised by a board-certified pathologist with training in molecular lab. Yan. So, kailangan ng pathologist. Sorry na ulit itong dalawa. Okay. So, what is the required physical facility by the AO14? Yan. So, medyo may pagka-inconsistent yung AO14. I'll tell you later. Okay. So, number one, sabi nila, dapat may specimen reception, a dedicated area where you will receive your specimen. Okay? So, this is a separate room. Okay? So, lahat to, dedicated space, separate room. Okay? Next, an area where you will prepare the specimen, the samples that you have received. So, ma'am, what type of preparation they do? So, ano ba? Centrifuge? Yan. What specifically? So, nucleic acid extraction. Hindi ba yan ang lesson 2 natin? And we inactivate the virus, especially if it's for COVID-19 testing. Yan. So, therefore, pag na-inactivate na yung virus, hindi na nakakahawa. Okay? Hindi na infectious yung virus natin kasi RNA na lang siya. So, this is the pre-PCR room. So, before performing your PCR. And we also have another pre-PCR room. Okay, maninil to ah. So before going to the amplification room, we have reagent storage or reagent preparation or handling or master mix preparation. So anything that is involved in preparing your reagents, aliquoting it, handling it, storing it, guys, is in your reagent. Yan. So dapat itong reagent area, sobrang linis na, okay? There should be no contamination, okay? Any amplicons, ibig sabihin yung products of amplification, dapat wala dyan, okay? Kasi nga, if it's contaminated, then it will make your results inaccurate. So, negative pala talaga yung patient dahil kasi sa contamination, naging false positive, di ba? So, dapat clean areas yung dalawang pre-PCR area natin, okay? Now, here we have the PCR amplification room. So, ang main na equipment dyan, hindi ba yung thermocycler and RT-PCR? If it's an RT-PCR facility. So, itong AO14, RT-PCR ang nire-record yan. RT-PCR, okay? Kasi nga COVID-19 testing yan eh. Tayo sa school lang tayo. So number 5, there should be an area for clerical activities, for correlating, understanding the results, and analyzing. Administrative work, dapat may hiwalay na opisina. Yan. So guys, ito yung color plan ng freestanding COVID-19 testing laboratory using RT-PCR. So, walang post-PCR area sa AO14. Okay? However, yan, may pagkalabo nga itong AO14, biglang sa ano nila, sa floor plan nila, may template addition room or template adding room. So, when we say template DNA, so the extracted from the sample is called your template DNA already. So, ito na yung ifo-photocopy nung RT-PCR, okay? So, it could be from the extracted DNA. Kunyari kung guys, bear with me. Kung COVID-19, hindi ba RNA virus siya? I told you, your thermocycler, guys, does not photocopy or does not replicate, make copies of RNAs. So what we do is we convert it into complementary DNA. So your RNA is converted into a complementary DNA. So they serve as your template DNA. Naiintindihan? Okay? So ito yung kokopyahin ng paulit-ulit. So iba pala yung room niya. Yan. Kasi nga, as much as possible nga, these are actually clean areas as well. No contamination. Yan. So dito paghahaluin yung template DNA at saka yung reagent. Yan, pwede. And 7, an area for changing. So there could be more than one changing room in a COVID-19 area. And we have post-PCR area. So in our laboratory in University of Baguio, we have post-PCR area because we only have thermocycler. However, if you are an RT-PCR laboratory, you don't need a post-PCR area anymore because the results will already be apparent ng RT-PCR. Andun agad yung result. Pero kasi pag thermocycler lang, guys, further processing is required. So, anong mga makina sa loob ng post-PCR area? We have electrophoresis, PCR, ELISA. Yan, sorry, PCR, ELISA yan. DNA sequencing. Yeah. So, this is considered a contaminated or dirty area. PCR room is also a dirty room. Okay? So, ma'am, di ba yun, may mga term nga tayo, dirty and clean room. So, there's such thing as dirty and clean area. So, the workflow in the molecular lab, guys, is a unidirectional workflow. So, ibig sabihin isa. It only goes from left to right, yan. Or right to left. Isang direction lang, hindi pwedeng bumalik. Backflow traffic must be restricted to minimum. You could not go back. So here in this image said by CLSI, so reagent preparation room, the master mix, they will give it to the sample prep or the nucleic acid isolation, then it will go to the amplification area. It goes only one flow. Okay, tinan nyo, may stop sign yung mga pabalik. Backflow traffic is not permitted because it will cause contamination. Because here in this area, guys, there are already 10 raised to 9. So, millions and billions of amplicons, nucleic acids are created already. So, this is a dirty area. So, notice guys, sa specimen prep at saka PCR area, dapat negative ang pressure. When we say the room has a negative pressure, the air stays there, okay? So, the airflow goes inward, paloob. So, negative pressure is good in areas where you want the substances will stay in that area. Hindi tatakas talaga yung hangin. So, hindi tatakas itong mga aerosol from bacteria, virus, yan, specimen prep. Dito rin, hindi rin tatakas yung mga amplicons, yung mga products of amplification. Yan. Dito sa reagent storage and master mix prep, positive pressure, okay lang umikot kasi nga, it's a clean area. Yan. So, itong negative pressure, if you want it to be talagang kulob, yan, isolated, contained, talagang contained. Yan. So, kunyari, sinabi mo, it's contained. Yan. It cannot go out. Okay. Yan. So, both cleaning personnel, and specimens should have a unidirectional workflow. Mga tao, one way. Mga naglilinis, one way lang. Mga specimen, isa lang. Hindi bumabalik. Okay? So, this one, itong reagent storage area natin, at saka specimen prep, these are amplification product-free product rich. Yan. Dito kasi nga, sabi 10 raised to 9 amplicons. So, contaminated talaga yan. Remove PPE before leaving one area. Avoid or limit reverse direction. Yan. As much as possible, avoid. And reusable supplies in the reverse direction needs to be bleached. Yan. 10% sodium hypochlorite. Ma'am, so ba't sa school, mga balik-balik tayo? So, that is just a study, molecular bio area. Yan, teaching molecular lab. Pero, yan, so magiging strict rin tayo dyan sa unidirectional workflow. Kasi hindi tayo kasya sa laboratory. Pag may holding area na tayo, siguro we would do that. So, features of each area. So, lahat to is dapat available sa lahat ng areas. So, each area should have separate sets of equipment and supplies. So, kung ref, sa specimen prep, it should stay there. We have pipettes, filter tips, centrifuge, timers, vortex, lab coat. Dapat nasa isang dedicated area rin siya. Cleaning supplies, office supplies, and ventilation system. So, there should be a good ventilation system on each area. Yan. So, these ones, itong laminar airflow and dead air box could be present in all rooms. Yan. So, laminar airflow and dead air box could protect the product from any contamination. However, it will not protect the personnel. Yan. So, if you want a sterilized area to work on, maganda ang laminar airflow or dead air box. Yeah. So when you say dead air box, the air is not circulating. However, merong ultraviolet to sanitize the area. Yan. So, tapos I'm searching, ma'am, ano bang may mga questions sa internet. Which one is better? Laminar airflow, dead air box, yan. So, depending on the need of the laboratory, yan, and the budget. However, they are good in ensuring that our products are not contaminated. So, you could use this in your reagent mix. You could use this in, while if you need an area where you could write, ganyan. So, what else? We have pass boxes, yan. So, in our school, right, ano lang siya, mukhang bintana na pang drive-through, yan. So, these pass boxes is an area, actually, to maintain a aseptic transfer of materials between two controlled areas. So, if you want to pass your extracted nucleic acid, yan, sa template addition, yan. So, dun mo ipapasa so that the personnel will not move around anymore. And we have the air pressure. Yan. So, the air pressure are important in containing the nucleic acid. Yan. Sa sample prep, post-PCR, negative pressure. Reagent prep, positive pressure. Okay, for the equipment, yan, mabilis na lang to. So, for the sample preparation area, so we need minifuges, yan. So, in extraction, di ba, kailangan mong i-separate yung DNA from the organic solvents. What is required according to AO14 is BSC class 2A2 to protect both the personnel and the product. This is an autoclave. It's a steam under pressure, 121 degrees Celsius, 15 psi. So, that's the pressure for 15 to 30 minutes. Yan. So, we could autoclave our glasswares. We could autoclave our culture media if you need one. Okay, what else? We have vortex mixer, pipette. Yan, pausahin na lang. Ito, bago, heating block. Yan. So, some kits, extraction kits, requires to be heated. We have cold rack naman. Yan. So this, the PCR tubes needs to be in a cold temperature. So yan, it will maintain the coolness of the tubes. So in the extraction of your DNA, yan, we need to centrifuge. So to separate the nucleic acid from the cellular debris, diba? Yung nucleic acid, it is supernatant, it floats. Yan, the lipids, it settles at the bottom. So, using minifuges is very important. So, iba naman yung kaso pag elution ginamit mo. However, we still need minifuges. Gagawin natin yan. So, we have master mix area. Yun. Sinabi ko na to, we have filtered pipette tips. Yan, to prevent carryover. So, para hindi ma-contaminate yung piston ng micropipette natin, we have laminar flow cabinet, we have PCR cabinet. Ayan. So, parang magkakamag-anak to, low laminar flow, PCR, tsaka dead air. Kasi, yan, it only protects the product. However, magandang laminar flow because I think it has a HEPA filter which filters the air out. This PCR cabinet and dead air box does not filter the air that is coming from the cabinet. Okay, in amplification room, yan, we have a microplate spinner to mix your microplates that you place in your thermocycler. Yan. So, thermocycler, so this is the one that is our PCR machine. Okay? So, it has a different varying temperature. Ano na nga ba yung temperature ng thermocycler? Guys, please, lalabas yan sa quiz. Yan. So, anong temperature ng denaturation? Anong temperature ng annealing? Anong temperature ng elongation and all? So, this is the RT-PCR, yan. So, hindi mo na kailangan, so sa RT-PCR, ang galing, lalabas na agad yung results kung positive or negative sa COVID-19, the number of cycles, yan. So, milyones yan, kaya wala pa tayo sa UV. Yan. So, post-PCR, so in AO14, walang post-PCR room, okay, sa atin lang. So, we will perform gel electrophoresis, yan. So, dapat magaling ka mag-pipette kasi ang late lang nito. Yan, we have gel documentation box. Ito nalito ako dito sa gel documentation box natin. Ang high-tech na kasi nito, guys. But it started just like this. Yan. Yung mga gel documentation box, it started as UV trans illuminators. So, pareho sila na may UV. Okay? This ultraviolet light will make your fluorescent stain more visible. Yan. We have post-PCR. What else? We could do DNA sequencing. Please read on what is DNA sequencing. PCR ELISA. Sobrang bongga nito. So, your clinical chemistry is combined with your, with clinical chemistry, immunology, molecular biology kasama na. Kasi papansin nyo may antibody reaction eh. Tapos may enzyme. Grabe, no? So, talagang ultra-sensitive na ang PCR ELISA if you want to detect a nucleic acid or a sequence of a DNA. Grabe. Yan. So, guys, that's it for our discussion. Thank you for listening. Bye-bye. Good night.